So yeah, I'm trying to not call it a mock exam anymore because I got a lot of feedback about this is not like an exam, which is true. It's meant to be more of a study guide to show you the types of concepts that will be tested on, but not necessarily the types of questions that will be asked of those concepts. So whether or not it's easier or harder than previous mock exams that I've given, I haven't written the test yet, so I'm not sure what it's going to look like. But it does, I am trying to correct a little bit to make, from feedback a lot of you have given me in office hours after the first couple of exams, I am trying to word the questions more clearly, maybe with less paragraph style, read all this and then answer the question. So in that sense, it might be easier, or more straightforward, let's put it that way. Not necessarily easier, but maybe easier to answer, I hope. That's my goal. I'm not trying to increase the probability of getting an answer wrong because I make it difficult to follow the directions. And so because I will be in class again this time during the exam, I apologize for having to have missed last time, do remember to please ask me questions about interpretation of questions during the exam if you have them. So that was a question about exam structure, content, sort of. Other questions about content or the exam? It'll be paper again, so I'll bring paper copies in case you want to do the paper version. Same submission process though, if you do the paper version, take pictures and upload those to the Google Classroom assignment. Yeah?
Will we have to use BLAST for this exam?
You will have to use any sort of techniques that we've studied since then. So was that on the slide I put up at the end of last class that suggested, or was in last class's slides? Let me check. I think I listed it. So it's possible. You'll have to do sequence alignments, BLAST, know about accession numbers. Now's a good time to ask related questions if you want. Yeah.
Could you possibly go over one more time how to look up a BLAST sequence?
Sure. Let's see. I'm just looking at the list from last class. So this, let's see. So yeah, I'll be asking you to do potentially chi-square tests. So you need to know how to do the chi-square formula. You can do it on a calculator, you can do it by hand, you do it on a Google Sheet or Excel spreadsheet, whatever you feel more comfortable with. Let's see, use of Venn CBI database and BLAST, so we'll definitely talk about that right now. Again, ask me questions as we go. So that includes knowing about accession numbers, how to access the sequences of genes, conducting and analyzing pairwise sequence alignments, calculating percent identity between sequence alignments. We did have one case study paper. We haven't done much yet in transcription initiation, mostly the concepts of necessity and sufficiency, reporter genes, and then using multiple sequence alignments to identify potential transcription factor binding sites. Oh, well, that's convenient. What would you like to know about BLAST, accession numbers, binding gene sequences?
What's the difference between a subject accession number and an accession number? Are they the same thing or synonymous?
So the question was, is there a difference between a query accession number and an accession number? And the idea is that when you perform a search, so this part only applies to BLAST, what I'm about to say. There's the query sequence, which is the sequence that you are pasting into a search field. That's the sequence of the gene you want to find matches of. So you think about that in terms of any sort of search engine, that's the search term. It's what you're looking for information about, and subjects are the responses. Search results. So in this case, the search results are DNA sequences that exist in whatever database you're looking at. And accession numbers apply to all subjects. So every subject, every entry in the database every possible subject has an accession number. That's its unique identifier. So it's like street addresses for houses. The house number, you've got the street name, that's a unique identifier to a particular place. Every DNA sequence that's in the database, every DNA sequence record, like the sequence of frog DNA polymerase, has a specific accession number that you can use to look up just that gene. So then I think that leaves us with, do queries have accession numbers? And the reason that I'm hesitating there is because there are two different types of ways to do queries. So if you open the BLAST, nucleotide BLAST tool, you get the search window, and it says you can enter accession numbers, GIs, which is another form of accession number you don't have to worry about, or FASTA sequences. So if you can enter an accession number. If I say here's an accession number, blast it, you can type the accession number in here, and that does the same thing as entering DNA sequence into that window. So you can either type in sequence like I have, that's one way to do a blast. Or you could just type in the accession number of a gene. So the point being, this sequence does not have an accession number. This is a query, but it's a gene, it's a sequence I made up. Or if this was an exam, it would be a sequence I'd give you, and I say, here's an unknown DNA sequence. Unknown means it doesn't have a record in the database, so it doesn't have an associated accession number. It's just a sequence. If it's a gene that's already in the database, then you could search the database with that gene sequence as the query, and that's where you could paste an accession number in here and use that instead. Does that clarify that enough? Follow-up questions about accession? Yeah.
On the practice exam, on number 3, you gave us an accession number and like asked the name of the gene.
Yeah.
For the accession number, would you just paste it in the?
You don't, let's see, yes. If you start with an accession number, the easiest way to find out about that is to do an NCBI search. And I'll show you what I mean by that in just a minute as soon as this is done running. Because what BLAST does is it uses that query sequence to look for other sequences that are like it. That's not the best way to find out information about that query sequence in particular. The best way to do that is to go not to here, but just to the NCBI website, so I can use this link up at the top banner to go to the root NCBI page, and that gives us this search at the top. So this URL is just ncbi.nlm.nih.gov. And it will provide all URLs that you will need as well, so you don't have to remember any of this. But of course, it's open note, so if you want to take a note now, that's fine. And I'll write it down on the recording too. ncbi.nlm.nih.gov. So if I had an accession number, like that one, NG_011618.3, I can search there, and it gives me a summary, and it says, well, look at that, the top thing. It gives you the information about that accession number. Species, gene name, and you can click on that for more detailed information. So blasting is more often used to summarize, to find out where an unknown sequence is from. So it's more typical for a geneticist to paste a DNA sequence into the search window on BLAST and to find out what comes up when you search what kind of DNA is this that I just pasted the sequence of into the BLAST window. This is incredibly long. It's taking a really long time. Maybe something weird happened.
When I tried doing that one at home, it just kept timing out.
So is the take-home message that I should not have you rely on using BLAST during an exam, or should I'll have a backup. I'll have a backup option in case something weird happens to the network during an exam.
I was gonna say, I don't think it's gonna work for the blast part, cause it spent like 10 minutes doing it and it never loaded.
Yeah, I tried it like 4 times and it never did.
There might be a temporary issue with Blast too.
I think it only works doing that.
Because it worked when I did like the nucleotide sequences, but when I did the accession number, it didn't work.
How weird. Well, maybe that Blast is temporarily broken. It's worked for me in the past, but if it's not working now, then we won't use this now. Which is totally fine. And that's why I like having these little practice sessions, because we can run through things and make sure that things are working. Other questions? Yeah.
Number 5, part B, like I understood part A, but then you.
At the bottom, how it's like the new.
Okay, good. I was looking for an example accession number. So again, using this website, accession number goes in there, so I'll type in ng underscore 001618.3. So there are a few options that we've played with in the Blast window before. We always use the search window, of course, and then there are all these different things you can customize down here. And I'll tell you explicitly which one to use if you have to do Blast. The only two we've really used are the Human Genomic and Transcript Database and the NRNT database. This is the default, and that's, if I don't specify, you just use the default, which is the NRNT database. If you paste that in other words, this is a complicated web page with a lot of options and features, but we haven't talked about any of the options and features. So I hope they're not distracting, but don't worry about all the rest. If we haven't talked about it in class, this is the only thing we've done is change the database and work with the search window. And presumably, if I hit Blast, it will take that accession number, and I must have typed it in wrong.
011.
011, thank you. Try that. And sometimes it takes a while for it to search the billions of sequences in the database. Wait for it. Any other questions I can answer in the meantime?
When I did it, I didn't go through Blast. I just used the search bar at the top. Is there a reason we shouldn't?
Like
do
a
that?
web search engine?
The NCBI
Oh, okay.
search engine.
Yeah, so you could do that. So it's another good point. If you have an accession number,
Do you have to change like the setting type thing or the ref seek? Does it matter which one?
excellent question. So, which question was that on the mock exam?
Number 3.
3. Did I go past it already?
Species, and I'm not quite sure how you did that. Like how you searched it using that information given.
Okay, what species is the query species from? That's the mock exam or study guide, whatever you want to call it. Question 5. I give a sequence, you calculate the percent or the percent identity, and then what sequence is that from? Let me see. What I did was I just copied and pasted that DNA sequence and blasted it. So let me do that.
Was it the top line or the bottom line?
Does it matter? I would have used the top line because that's the query, that's the thing I was searching with. But what do you think would happen? I mean, it's pretty similar to the subject sequence. So presumably, if you used either of those sequences, you'd come up with very similar subject results. Because they're very similar sequences. They should match very similar genes in the database. So that is what I did. Thanks for the feedback on Blast not working too, that's good to know. So we'll be only pasting stuff in. Other questions? And the reason that I like to distribute these exams as PDFs, of course, I think it's of course, because when you can double click and copy and paste sequences, it's faster than just having to type them out by hand. So hopefully you can take advantage of that. One reason, even if you use the paper copy.
Can you go over how to read the gels for number 6?
Number 6. And what was it? I believe I've skipped answering somebody's question to the fullest extent of my ability. So if you did ask me a question and I haven't answered it yet, ask again, please. Question 6. What were the questions about question 6?
The figures below are similar to, but different from the SRY cisgenesis from Koopman et al. In 1991. A, in the figure at left, what are the chromosomal sexes of 3 mice? B, in the figure at right, suggests one genetic reason why the sex phenotypes male or female of the 3 mice do not agree with the gel evidence of the presence or absence of the SRY gene in those mice.
Okay, thank you. So the panel, what I got out of that question was, on the left, we're trying to figure out what the sexes of the genetic sexes of the mice are. And on the right, there's going to be something that suggests there's a disagreement between the phenotype, that is the physical sex, the external sexual genitalia of the mice, and what the genotype indicates. So what's the one piece of information we use to tell the genetic sex of these mice using PCR? This is PCR for 3 different genes: SRY, a control gene, and ZFY. So what's SRY? That's the male sex determining gene in mammals, mice, humans, other mammals. So should there be perfect concordance between the presence or absence of SRY and the sex of the animal? What's the pattern between the presence of SRY? Band means that that animal, 33.17, has SRY, doesn't have SRY, does have SRY. So what's the pattern?
The patterns have SRY, it seems like they're male, phenotypically.
Okay, so every time there's an SRY band, that mouse has external male genitalia. That's correlation. It is actually causation. The presence of SRY is why those mice look like they're males. They have the SRY gene, they develop as males. And the one mouse that doesn't have SRY is a female. This is evidence that SRY is the sex-determining gene. There's a perfect correlation between the genotype of the mouse and its phenotypic sex.
What is the M bar on the left?
Marker. DNA ladder. So this is a set of bands of known molecular weight. And that's just so that the scientists can assess the relative size in terms of number of nucleotides of the bands. And they have not put here on this, in this paper, they didn't put the sizes of those bands, so we can't do anything with that information. But we did work on molecular weight ladders on the previous exam. I had you draw out a gel. The restriction digest, the last question, the one that was a really hard question, or draw a gel of what would happen before and after digest of something. And on that question, I had a molecular weight ladder like this one, but I had listed the band sizes because it was necessary for you to know to put your PCR product at the right molecular weight. Here, they haven't told us anything about the band sizes. It's just a standard. And the positive control is present in every sample. That basically just tells the scientists that they actually did successfully add DNA to all of these PCR reactions to amplify something. And what's ZFY?
It's related to SRY.
It is related to SRY, but in an important way. How is it related to SRY, aside from the fact that the third letter is also Y? Right, they're found on the same chromosome. They're both Y-linked genes. But it's not a gene that's involved in sex determination. It's just another gene that's present on the Y, and it's also not on the X. So specifically, it's only on the Y. So how did the scientists use that piece of information? What did that tell them about these mice? And the genetics of the mice?
There's further evidence that they were going to be phenotypically male.
It means that those mice not only have the SRY gene, but they have a Y chromosome. Both of those genes are on the Y. So SRY is on a Y chromosome. That's true. Some of you are looking kind of shocked. We have two pieces of evidence up here that these mice have Y chromosomes. Presence of SRY, presence of ZFY.
Can you explain the significance of the control band again?
Yes. So significance of the control band. Just a sec. Fine. I was trying to duplicate this slide so that I could draw on it again, but I guess I will erase temporarily. Well, okay, let me finish this question first, and then we'll do the significance of the control band. Why do these scientists need two different pieces of evidence, presence or absence of SRY and presence or absence of ZFY, to determine whether or not a mouse had a Y chromosome? In the original experiment, yeah.
And if the mouse didn't have the Y chromosome, but it was phenotypically male, then it was proof that the S or Y.
So that's why they need this information. We've got SRY present in both of these mice, but that could either be because the mouse has a Y chromosome or because the authors put SRY into the mice. So they have to have this second piece of evidence to show that those two mice do actually have Y chromosomes, and that's why they're males, because they have SRY. So control experiment. What's the control experiment about? Always have to do control experiments when you do PCR. What would happen, how would this be interpreted, I suppose, if, I know I'm coloring in a darker black than necessary, but what would happen if that gel looked like that? Where the female mouse had no bands for any of the genes that they were looking for. There are two different possibilities. What's one?
The gel failed.
Okay, that's the most, something failed. That's why this is the positive control. It's to detect whether if something went wrong and nothing worked. But what would be the other interpretation of a mouse that has no, if we didn't do that experiment at all, if they just looked for presence or absence of SRY and ZFY, if there are no bands, what does that mean about that mouse? Doesn't have, you'd interpret that they don't have SRY.
No, SRY or no ZFY.
So what would the conclusion about it, if we were just thinking about, okay, what's the genotype of the mouse? It doesn't have SRY, it doesn't have ZFY, what would that mean?
You need both to be
It's,
male.
it's a female mouse. There's no evidence of a Y chromosome. So that would lead you to conclude this is a female mouse. But we don't know if that PCR worked, because the other interpretation of a totally blank well is PCR failed. That is, you forgot to, not you, it's not your fault. These authors forgot to put the TAC, the DNA polymerase, in the reaction, for example. That's another reason why there would be no bands. So it could be, in that case, it could be that that was a male mouse, but the reason there are no bands is because they forgot to put the primers in the PCR reaction, or they forgot to put the tack in the PCR reaction, or they pressed start on the PCR machine, but there was a power failure, so the PCR didn't actually happen, and they came back the next day and analyzed the data, and nothing happened. So you have to have the positive control because you need to know that PCR worked for some gene in this mouse, and then, and only then, can you evaluate the fact that there was absences of genes as evidence that those genes themselves are missing, not all of the DNA of the mouse is missing or that the PCR didn't work. So that band there is critical. It means PCR worked for that sample. Therefore, the absence of those bands means that those DNA sequences are not in that mouse. And then phenotype and genotype disagree. What doesn't make sense? How do the genetic and phenotypic data on the right side not agree with each other? The DNA genotype data and the phenotype data. What's wrong?
So wells in 2 and 3 look identical in this gel, but they have 2 different phenotypes.
Okay, right. So the pattern is the band pattern is the same, but the phenotypes are different. One's male, one's female. So that's the problem. Then the question asks, provide one possible explanation for why that could be true. So which of those lanes, the female 33.9 or the 33.13, which of those makes sense in terms of the genotype and the phenotype?
33.13.
And that's because it's male and it has SRY. So should it have ZFY?
Yeah.
It doesn't have to if it was.
Yeah, if it was, I think, that thing. Yeah, if this is one of the transgenic mice, that's an XX mouse that the authors put SRY into, it would have no Y chromosome, but it has the gene and it developed as a male. So that is straight up from that paper that we talked about.
That's a genotypically female, but phenotypically male.
Right, it's genetically female because it's got no Y, and if it doesn't have a Y, then it has to have no Y, therefore, ... has to have 2 X's. But it developed as a male, and that's because SRY, the addition of SRY caused male sex determination. So the one that doesn't make sense is 33.9. Doesn't have a Y chromosome, there's no ZFY band. Does have SRY, so it should be what sex? It's got SRY, it should be male, but here it was a female. Of course, I used Photoshop for this. This is not real data. Don't go home and tell somebody that this is real data. I created the figure on the right. So what is a possible explanation for the fact that this mouse has the SRY gene but is female? This is where you can be as creative as you want. It has to be a scientifically rational creative, but
There is a gene that is not on this chromosome that determines female development.
well, we know that SRY is the gene that turns on male development. So if it's got the SRY gene, you would have to hypothesize that there's a female determining gene that supersedes or that overrides SRY.
Or coincides.
Oh, fair enough. Other thoughts?
It could be that they accidentally added the contents of 33.13 to 33.9. And that's how they get the exact same result in the last few lanes.
Okay, so that is creative answer. Not what I was thinking of, but I would give you points for that if you put that on the exam, if this was an exam. The argument here is a technical or an experimental one, that whoever was actually if I understand correctly, that whoever is loading this gel loaded the same DNA from mouse 33.13 in that well, which is why they look identical, So there is no DNA from this female mouse on the gel, and if they had actually taken her DNA and loaded it, it would have looked correct. That's it. Sure. That happens a lot in running PCR reactions on gels. Accidentally skip a lane or add the wrong DNA in the wrong well or something like that. I think the one that I put on the key was they might have, this SRY gene might have a mutation in it that prevents SRY from doing its job in turning on male development, so the mouse developed as a female. So the gene's still there in the genome. It's able to be PCR amplified, so we can see a band, but what the band doesn't tell us is whether or not the gene that's encoded by SRY is a functional gene or some sort of weird mutant. That's just one possibility. Another possibility, just off the top of my head, might be that if this were real data, that they had injected that gene into this mouse and they took those cells from, that do have the SRY gene from like the ear of the mouse. It's common actually to clip mouse tails to get DNA from mice. You just cut off the tip of the tail and extract DNA from it. That way you don't have to kill the mouse to get DNA from it. But does the DNA that's in the tail itself have anything to do with sex determination? It's not something we've talked about in class. So I don't want to distract you too badly, but where in the body do you think it's critical to have the SRY gene turn on to start male development?
Autosomal cells?
In the head? An arm?
That's a word.
The gonads.
Yes, one of the places is the gonads. So who cares if this, so this is just another example. Maybe these cells are from the tail of the mouse, but who cares if SRY is in the tail of the mouse? Mouse tail has nothing to do with sex determination. It had to be in the specific types of cells that initiate sex determination. So those are all possible explanations for that.
The mock exam didn't have anything about the chi-square stuff. Is that gonna be on the exam?
Very likely. Well, it's on the list, right? It's the top thing on the list. Chi-square, etc. So if I didn't put an example of that on the mock exam, so to speak, because I had passed out a bigger Google Sheets-based version earlier in like the week prior, I think. And I can't ask questions about everything. So of course you're going to spend lots of time studying some of these things that are not going to be on the test. Unless you want to have a longer test. I could represent everything that's on there so that make sure that everybody, whatever you spend your time studying is guaranteed to be on the test.
Or just, okay, sure.
I would be like the least popular professor ever. Yeah,
How many points is this exam worth again?
15 points. There's a 15 point, it's the last 15-point exam. So that's 3/4 of a letter grade worth of points.
And that makes the final worth 5?
And the final's worth 5 points. So some of you have already been to office hours and we've discussed this a little bit, but the math goes something like, you need to get, let's take the example of a student who wants to get an A. I hope that all of you want to get A's and earn A's. I do hope you all earn A's. So you have to get 80 plus points by the end of the semester. So let's say, hypothetical situation, let's say right now you've got 55 points. I don't even know if that's possible yet because there's a lot of points left to hand out, to assign. So 50.5 points now is between 40 and 60, so that's C. That's a high C, C+. If you got 10 points on this coming exam, that would put you at 65 points, and that's a B. So if all you wanted to do in the class was to earn a B, and that was fine by you, then what happens if you don't come take a 5-point final? You don't have the opportunity to earn those 5 points, but you've already earned 65 points. Sorry, I'm not projecting this. So you get a B. So if by the end of the semester, in other words, if you've got 80 plus points and you wanted an A, you've earned the A, you can decide whether or not you want to take the final, because it's only worth 5 points. If you find yourself in a point situation where you're on the borderline of getting up to the next higher grade, that might be a really good reason to take the final, because those extra 5 points, not extra, those 5 points might pull you from like a 69 or 79 to 82 and cross the border from B to A. So it'll be really incumbent on you, I think, especially after this exam and after a lot of the assignment and quiz points for the rest of the semester we do over the next 2 weeks or so, to go into Grade Center, whatever they call it on Canvas, gradebook, and total up how many points you have. Don't look at the percentage, just go through every, I find it annoying that Canvas doesn't tell you how many points. It's all based on percentages. Total up how many points you've earned. Look at the syllabus and see where you're at.
Well, whenever I look at my Blackboard, it doesn't seem like all the points are up there.
That's correct. I've only added all of the assignments up to this point, and that's why it's confusing, because you might have 100% of all of the work that's been assigned to date, but it doesn't tell you how many points you've earned. You have to go through and tally those individually for all the assignments.
I tried doing that and it didn't add up to 56.
Okay, I'll take a look. Appreciate the feedback. But that's the approach that I would advocate using. Yes?
Can we go over number 9 on the mock exam?
Number 9. Right.
Injected it.
So in that study, with the cute little mouse in its nose. Yeah, I'm doing a pretty good job with art today. Great. They had a mouse that was, well, they were injecting the SRY gene into embryos. They're making cisgenic mice. They were experimentally putting SRY into mice that some of them were female, some of them were going to be male because they inject them as embryos. And so they were looking for mice that had no Y chromosomes, but had the transgenic, cisgenic, the added SRY gene. So these mice, if you drew this gel, would look like they had the SRY band. These mice have SRY, but do they have ZFY? No. There's no band where you'd expect to see the ZFY band. There's a blank there. There's a gap where I'm trying to draw a dash circle, but big fingers. That tells us that this is an XX mouse genetically, and then the authors look at the gonad of the mouse and discover that it's a male. So the reason that the authors need 2 pieces of information about sex, about the presence of the Y chromosome, is that in this case, the mouse has SRY, but not because it's got a Y chromosome. It's because the authors put that gene in this mouse. They have to rely on presence or absence of ZFY, which says that there's either a Y chromosome present or no Y chromosome present. And if there's a Y chromosome, that means that the mouse is XY. And if there's no Y chromosome, by default, then it has to be an XX mouse.
Wait, by no Y chromosome, you mean by no ZFY?
Correct. So ZFY is, they're looking at one spot on the Y chromosome. They're asking, is this part of that of the Y chromosome present in this mouse? And if it's not there, then they conclude that there's no Y chromosome. Yeah. See you next time. Yeah. See you next time.